Warahmatullahi wabarakatuh. Good morning, everyone. In this video, we are going to have a look at DNA and RNA structure. So now we know that DNA or deoxyribonucleic acid is the hereditary material in humans and almost all other organisms. Some organisms also actually have RNA as their hereditary material, but we're going to talk about that as well at the end. So DNA and RNA, it has the code, they actually bring the code for making proteins. DNA, this is what we typically, when we say DNA, this is what we think about. So DNA has repeating subunits or monomers called nucleotides. So this is one monomer, one nucleotide. Nucleotides can be divided into three components. So we have a phosphate group, a pentose sugar, there's five sides, a pentose sugar, and also our nitrogenous base. So for DNA, there are four major nucleotides based on their bases. We have the adenine, thymine, adenine here, adenine, thymine, and guanine, cytosine. So if you look here, they can be categorized according to how many rings are present. Double rings for purines, so these are the purines, adenine and guanine, and single ring for the pyrimidines. So here thymine, cytosine, and also uracil for RNA. So if all three of these components are present, we call it a nucleotide. But if it's only the sugar and the nitrogenous base, we call it a nucleoside. Okay. Here we have DNA and RNA. So DNA and RNA, the major difference is in the pentose sugar, where DNA consists of deoxyribose, pentose sugar, and also RNA, there's a ribose sugar there. For DNA, the bases would consist of AG, TC, while in RNA, the bases would be AG, C, or U. And then we have, so this is a singular nucleotide. So these atoms here, O, H, these atoms here are found within individual nucleotides. But they are removed when they join together to make the strands of DNA or RNA. In a DNA strand, nucleotides are covalently linked together by phosphodiester linkage. We have the phosphate connects the fifth carbon, this is the fifth carbon, of one nucleotide to the third carbon of the next nucleotide. So you see 5, 5, 2, 3. There's a directionality to the DNA strand. In the strand, all the sugar molecules are orientated in the same direction. The phosphate and sugar molecules form the backbone. This is the backbone of the nucleic acid strand, and the bases project from that backbone. No the DNA structure, three-dimensional structure, is a double helix, and these are the general structures, structure features. We have the two strands twisting together here around a common axis, where there are 10 bases. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 bases and 3.4 nanometer for one complete turn of the double helix. And the two strands are anti-parallel. Anti-parallel meaning that one runs in the 5 to 3 direction and the other runs to a 3 to 5 direction. The helix is a right-handed helix, and the double between the two strands, the structure is actually stabilized by hydrogen bonds. So look at the complementary bases. So you have your A and T. It's stabilized by 2 hydrogen bonds. C and G is stabilized by 3 hydrogen bonds. And the turning of the double helix, there are actually two asymmetrical, not symmetrical, grooves on the outside of the helix, where there's a minor groove and also a major groove. So don't ask me, it's something mathematical. And in terms of the double helixes, they can actually form different types. So there's alternative types of double helices. Most DNA in living cells would be a bDNA structure. This one would be this one, bDNA. However, under certain in vitro conditions, scientists has found these aDNA and also what they call zDNA structures. So the difference would be in terms of the right-handedness. So So in terms of aDNA, it's right-handed. So it's a right-handed, and in terms of facts, 11 base pairs per turn, and it actually occurs under conditions of low humidity. For zDNA, it's actually a left-handed helix, also 12 base pairs per turn, and the conditions are more concerned salt concentrations. So it can actually different in forms in high salt concentration and also in low salt concentrations. And then we have our RNA structure. So ribonucleic acid. RNA is typically single-stranded with ribose sugar. I did mention earlier that's the major differences with DNA. And you can see here, in terms of base pairing, A to U and instead of a T, and C to G. But the linkages are still the same in terms of a phosphodiester linkage 5 to 3 directionality, and having that phosphate and sugar backbone with the bases sticking out. So RNA can form, is typically single structure, but RNA can form short double-stranded regions as well. Um, so this secondary structure is due to complementary base pairing. Um, complementary complementary regions are held together. This is one strand. They actually fold together and form secondary structures. So because of that hydrogen bond, so you have your positive base pairing there. This is complementary regions held together by hydrogen bonds, like I mentioned earlier. And then in regions where it is non-complementary, like here, they have bases that are actually projecting away from the double-stranded regions. So you would get all these different types of secondary RNA structures. So this one is called a bulge loop here, complementary, non-complementary. This is an internal loop. This is a multi-branch junction. So, and also the stem loop here. This is your complementary region, and here would be your non-complementary region. So there you have it, the DNA and RNA structure. So these DNA and RNA structure are very long strands of genetic material. Imagine these strands to be very, very long. So to fit within a living cell, the DNA double helix must be extensively compacted into a 3D conformation. That's our chromosome. We will discuss about this in our next chapter. So that's all the time we have for today. So see you next time. Bye.